broadcast before, my name is Megan Peters. I am the president of Neuromatch Academy, and I am here to introduce you to Nici Pfeiffer and to Mark Musen, who, I hope I pronounced your names okay, who are going to tell us about the Center for Open Science, which is fantastic, and also Cedar and how they can maybe work together. And so I will let them say a few words to introduce themselves, and then we'll just go straight into the presentations. So Nici, do you want to go first?
Sure. Hi, everyone. Nici Pfeiffer. I'm actually the director of product at the Center for Open Science. It's my job to really work with the communities of researchers and stakeholders to understand how the infrastructure can support open science and the culture change that that would sort of be the thing we're all after. So I'm excited to present a little bit to you and also to start to hear your questions and get your feedback.
Great, thanks. And Mark?
I'm Mark Musen. I'm on the faculty at Stanford. I direct the Stanford Center for Biomedical Informatics Research. I have lots of interest in semantic technology and open science, and really excited about being able to think of ways in which the work that we're doing at Stanford may relate to what COS is doing. You'll hear all about that.
Cool, thanks. So I think you guys are going to do like about 20 minutes each, 25 minutes each, and then we'll save some time at the end for questions. Is that right?
That's the goal.
Okay, so all throughout, as usual, with all these Q&As, if you guys have questions while the talks are going, please use the Ask a Question box. If this is your first crowdcast, some of you may be new here. So if this is your first crowdcast, you've got an Ask a Question box at the bottom, and you can type in your questions there. So all right, so without any further ado, why don't we go ahead and get going then? And I will make your screen big, Nici, and you can just take it away.
Okay. Thanks, Megan. Like I said, I'm Nici Pfeiffer. Mark is also the colleague joining us from Cedar that will be co-presenting some of these slides, but we decided it was easiest to sort of break it up into two sections. So I want to talk to you first about the tools that assist with open science, making experiments reproducible and their data reuse. And the tool I'll definitely be mentioning most is the OSF. But a little bit of background about where I am from. I'm from the Center for Open Science, where we have a mission to increase the openness, integrity, and reproducibility of research. We're a nonprofit. We have open source software, and our tool is free for researchers. Along the bottom, you'll see some of the different foundations and government agencies that have an interest in the infrastructure that we've built and that are funding a lot of this work. Our organization is sort of divided into three main pillars. Policy is an area where we focused some staff and some efforts to work through the incentives to embrace this change of openness. We also do our own research to gather evidence of the change. And as I mentioned before, we actually build and maintain our own technology to enable the change. So when you look at this curve I'm sure you've seen before, how open science adoption and culture change actually takes place, where you have your innovators, your early adopters, then your majority, late majority, and your laggards. So that bell curve is sort of how this transition and this culture change actually takes place. The Center for Open Science has actually looked at this, flipped it on its side, and managed to put against it all of our areas of effort. So when you look at that innovator group, first thing that really is required is to make it possible. So that's where the infrastructure that we've built and maintained, the OSF, comes in. Then the next step is to make it easy. That's the user interface, and that's the effort we really put into user experience and the user research, getting feedback from the community, which makes it the next thing, which is communities, and making it a normative behavior, making it rewarding with incentives, and then making it required with the policy changes. So when we think about making it possible and making it easy, we really want to enable open behaviors to advance science. So when you think about open, rigorous, and transparent, those are the three sort of core pieces. And when you do that and you think about the outcomes, the advancing of science, we're looking at data sharing, data reuse, reproducible data, and even thinking about a stretch goal here of null findings. When you say, so much of the data, that people are putting out there, it's usually proving a hypothesis correct, but sometimes experiments don't go the way we think they will, and we have null findings, but that's actually the most valuable data we could share, because now we're saving funding and resources and time by learning those lessons and building on those lessons learned in the future. So if we have the infrastructure to share and reuse data and policies that require researchers to share their data, why don't we have loads of reproducible data? That's the question. And I will posture that I think this is because so much of the time we're focused on the wrong end of the workflow. We know that the incentives are for individual success around getting published, not always about getting it right. Saying that people aren't trying, but this is a certain stance you can start to think on a little bit. So if we really want to solve for reproducibility, we have to start with the end in mind. We have to show our work, share our work, and make sure we share all the work. So I think that's really what the OSF offers as a free tool out there for researchers. And I think coupled with Cedar and Mark's work, you can really look at good description through metadata to make that data really reusable and then reproducible, because you're really describing it well. So all of that sounds great, right? But I know incentives, time, gaps in the infrastructure, it's overwhelming. You don't know where to start. I wish I had a time machine to go back to the beginning, but I'm already in process right now and I need help. We hear this a lot, and this is really where I love to spend my time, because it could be overwhelming, but it doesn't have to be. So when you think about implementation of open science best practices, it's not an all or nothing. There certainly is a spectrum, and that's sort of what this arrow is talking about. And some of the very simple things that you can do are just using an ORCID, making your materials fair, using a DOI so it has persistence, describing it with good metadata. You can go up this spectrum a little bit more, and I'll just hit on these really quickly. Having open data, using a data management plan where you have a data dictionary to describe your data, using good version history, not local machines, but actually a tool that really, versions all of the aspects of the work. Having open materials, so not only sharing data, but the protocols, the scripts, the code, the surveys, electronic lab notebooks, whatever tools are available for you to make sure that other materials that go along with that data are available for any researcher down the road that's exploring that data or reusing that data or reproducing that data. Then we like to push forward when we think even before data collection, looking at pre-registration. So the process where you would write out your hypothesis, your variables, what your pre-analysis plan is for that data, designing your study with rigor, and even potentially engaging in some review aspects at that stage too. And then of course there's preprints where we can accelerate the dissemination of these outcomes, get feedback earlier, and be more accessible to others around the globe. So all of these are ways to increase rigor and reproducibility of research. And like I said, the OSF is a tool for researchers. Here's a couple of projects that I stumbled on recently, just looking at fMRI data. And on the right side, you'll see a project that's active and ongoing, and it has a lot of files and activity going on. And on the left side, you actually see the pre-registration that took place before this data was being collected. So there are examples out there. If this is something that, like I said, you need help. I need somebody to show me how to do this. And if it's not easily accessible, you can always look around on the OSF or reach out to others. Okay. So I wanted to take just a few minutes before Mark shows you around on Cedar to give you a quick tour of the OSF. There's full support for the entire research lifecycle. So the OSF is designed to help with project management, collaboration. Obviously, that has gone up a lot in the remote collaboration setting that we're all sort of in. And we've seen a lot in really interesting use cases going on that we didn't intend in the beginning, but it's neat to see that the tool also does some of these additional things. And the tool does pre-registration and file management, easy for sharing outputs and meeting funder compliance. So the next thing I am going to, like I said, do is go over to the OSF, but I want to share this link with you. So you can go to the OSF and enter this 5-character part of the URL 8A4MT, and it's a public project where you'll be able to explore as I'm exploring on the screen. Okay, so I will be stopping my sharing momentarily, but hang on for just a second and I will be right back with a new screen for you. Okay, let's see. Okay, coming at you just a second. Sorry, it's loading.
It'll come. Don't worry. It'll happen.
All right, we are almost there. Okay.
Here we go.
All right, so hopefully
Yeah, perfect.
You can see my screen. So this is the public project that I created for you for the Neuromatch Academy 2020. And I wanted to just give you a quick tour, but also point out some of the helpful resources that I've put here. So after this session, even if you're looking at it as a recording, you can always go here and find more materials that might be useful. So a couple quick things. When you go to the OSF and create your free account, you can create a project. That's what I've done here. My project is private by default, but as you'll see up here, I have the buttons to easily turn my project public or private. So when you get to that point where you've curated this project, you've added collaborators, and you're ready to share it with everyone else, you can flip it to public. So I just wanted to point that out. Here you can see how easy it is to add your collaborators. So one of the things that I've already done is added Mark to my project. So you'll see him listed on this space, but it's really easy to search for new folks in your network. And add them, and you can type in their name. Let's see, I'm going to add my colleague from the center, Eric Olson, and so I can quickly find him. I know that's him, but if I wasn't sure, I have these additional links to go see his LinkedIn or his Twitter account or his GitHub account. Also, if he had an ORCID, I could do that as well. But I know Eric, so I know this is the right person, and I can decide what level permissions I want to get him. And we give you this really nice, easy sheet to see the different levels and what folks can do if you add them to your project. So definitely, look at that, pay attention. But I'm adding Eric. If I didn't find who I was looking for, it's really easy to also add them with just a name and an email, and they'll get an invitation to the project and to the OSF. But as I said, this is easy to add collaborators, and then every project has flexible structure. So you're looking at the top level project that I created, but over here on the right side, you can see this component section. I've actually linked some additional templates for you all just to have some more resources on how the OSF can help with research collaboration, it actually serves as a lab notebook or for course management type of use case. But I can also add a new component here very easily. I enter a title. So if I wanted to create my data component here, again, it would be private by default. And just because I make this top level project public doesn't mean that the data will be made public. And that's something that has its own permission, its own public-private setting. It also has its own set of collaborators. So for example, I might not be collaborating with Mark on data collection, only on design and the rigor of my study, but my data collection is going to be with another lab somewhere else. And so I would add those folks as collaborators, but I don't necessarily have to add them to all parts of my project if I don't want to. It's meant to be flexible to meet your needs. Also, another thing I wanted to quickly point out is add-ons. So the OSF actually has 11 different integrations with storage providers. This is so that we can meet researchers where they are. We understand that you get used to using a certain tool, or you only have access to one or two storage providers, just depending on your environment and the circumstances. And instead of having to move things and move data and reorganize everything, we want you to be able to quickly just integrate that right into your OSF project. So what I've done is actually done this in advance because there is a little bit of authentication that has to happen, and that can take a couple seconds, and I didn't want to slow things down. But I've authenticated in my Bitbucket account, and I've also integrated in my Google Drive. So you're looking at a public project where my files from another location are here, and you can view them. And then Mark, because he's also a read-write contributor, can actually add a new version to these files, and they'll be synced up with both of those external storage locations. So this can be really useful in just creating those good collaborative spaces with not a lot of burden. The other item I wanted to quickly run through with you all is that pre-registration concept. So within the project, you can click on this registrations tab, which is the beginning of starting a pre-registration. You will be able to click a button that says add new registration, and then you'll see a list of different registration templates. And that's a word that we use to just describe the different types of pre-registration that are on the OSF. And we're actually actively working with other communities to put in templates for qualitative, templates for existing data, for systematic reviews. So we work with communities all the time to develop these so that you can find what works the best for your methods or your specific research style. So if we just look at an open-ended registration, which is our most simple, basic registration, it basically walks you through with questions, and you provide responses. And then at the end, it creates this immutable, time-stamped archive of your project at that time. So what's nice about it is when you've set up your study design and your initial plan, you even might have your pre-analysis plan, you can upload that into your registration, and then it's captured there forever. It could be made public immediately, or you can embargo it for up to 4 years. So if it's something you don't want out there right away because you don't want your protocol shared at this time because you're still vetting things or whatever the case may be, it's designed to support that as well. So creating the registration, you add a little bit of metadata here, and then you go in, again, depending on the form, put a description in, and then you can upload protocols, surveys, whatever your instruments are if you want. And it's really easy. If I've already added it, then I can very simply find that file and select that as part of my registration. So that is my very fast tour of the OSF. There's so much functionality in here. So I encourage you to take time to explore and learn more about what the OSF has to offer, because I think it could really help with the research workflow that you're, I'm sure, spending your days and time in. I just have 1 or 2 more slides that I'll quickly do, and then I'll be handing off to Mark. Again, that link is in the slides, which I didn't point out, but they're actually uploaded in that project. So Mark's slides and my slides are both there in case you want to go back and refer to anything that either one of us mentioned. Let's see. So I think my slides are shared. Okay, good. So I just want to go to this last slide, which is just sort of recap of everything I was saying, which is obviously the sharing of open data is a fantastic thing. And I'm delighted to hear so many researchers are really thinking about this now. However, open data is only really as good as all the other things that you provide with it. So the metadata, a strong data management plan, all these other things. And so I like to think of it as an iceberg. And so you have the iceberg at the top, you have this data, but you really want to look below that waterline and think about all the other things like a good data dictionary, your analysis code, all these other things that took place that really support further exploration of that data, reuse of that data, and really replication of that data. So those are things to keep in mind. This is, for me, where I always kind of like to come back to. Okay, and that's all I have. and I'm going to let Mark go ahead and share and introduce sort of his section of this.
Great. Thank you, Nici. That was great. We've got some questions too, and we can save them for the end. But while Mark is getting set up, I will remind everybody, please, oh, now we have like the infinite, spaceship here. But please ask questions and put them in the ask a question box if you have them as we're going along. And then we'll have some time at the end to answer some of these questions as well. So I just put one in based on the results from the polls. Thanks everybody for doing the polls who's here. But it looks like we've got some questions now about pre-registration. So maybe we can get to those at the end and talk about those kind of after we see the Cedar platform as well. So, okay, it looks like we are all set up here. So yeah, please, Mark, take it away.
Okay. Well, I'm going to tell you about another tool, which I think is very complementary to what you've heard about OSF, and that's Cedar. Cedar was developed by the Center for Expanded Data Annotation and Retrieval at Stanford. And Cedar is all about creating better metadata. And the metadata component has already been alluded to, but I think I want to really emphasize why metadata is so critical. And it is a little bit different from the issues related to rigor and reproducibility that Nici talked about, more in tune with the importance of being able to re-explore legacy data sets and to make new discoveries from existing data. And to clarify this, I think the best thing I can do is tell you a story. I want to tell you a story about a colleague of mine on the Stanford faculty whose name is Pravesh Khatri. Pravesh calls himself a data parasite. And what Pravesh has done throughout his career is to go out to the publicly available repositories of data, typically mandated by the National Institutes of Health of the United States. And what Pravesh does is to look at legacy data that are sitting out in these repositories, contributed by other investigators, and to see what he can find. It's not quite so random. He actually has hypotheses in mind. But what he does is he looks at legacy data, and he's made some absolutely remarkable discoveries, spending a lot of time looking at GEO, the gene expression omnibus, which is where all the transcriptomics people store their data. And he's identified genomic signatures for diseases such as incipient sepsis, for TB, for being able to distinguish viral pneumonia from bacterial pneumonia, genetic signatures for identifying rejection of organ transplant. So he's now working on COVID, not surprisingly. Here's a guy who's made all these great medical discoveries, started 2 companies, we're using these biomarkers for really great stuff, never touched a pipette, never seen a patient, and just been enormously successful. And he's been enormously successful because he's really, really good at doing something that is really, really hard. And that is looking at legacy data sets and trying to figure out what the investigators are trying to do in their studies, figuring out what they actually meant, figuring out what the data actually are all about, and using those data for testing these additional hypotheses. Now, the problem that Pravesh has is that it takes a lot of work to be a data parasite. In fact, his postdocs and his graduate students will spend weeks going through publicly available data sets after what you would think would be a trivial search to find the data sets that are going to be supportive of his particular hypotheses. And what he's identified is that you can be a data parasite only if the data are accessible in some repository, if they're findable through some sort of a search facility, and if they're self-describing so that third parties can make sense of the data. And that means self-describing not just in terms of the characteristics of the data values, but understanding the experimental procedure and the experimental conditions under which the data were collected. And basically the data have to be collected with the idea that the data are going to be reused, that they're going to outlive the experiment for which they were collected, and indeed that the data are going to outlive the experimenter who collected them. But the real problem is that when you go to public repositories like the Gene Expression Omnibus and all the other NIH data sets and everything else, the data are a mess. As you are obviously aware, investigators view their work as leaving a legacy of papers, not leaving a legacy of data. And as those of you who are experimentalists know, creating metadata to describe data sets is just really, really hard. If you're creating metadata to describe a microarray experiment whose results are going to be put in GEO, they throw at you a spreadsheet. and you fill in the spreadsheet, but the spreadsheet really doesn't tell you what you need to say. It doesn't really tell you how to identify the metadata terms and to fill them in using terms from controlled terminologies and ontologies. Doesn't tell you very much, and it's really confusing, and not surprisingly, most people do a terrible job. Not because they're being sloppy or because they're trying to get through the task quickly. It's just that it's just hard to do this. And what that means is if you're a guy like Pravesh and you're trying to search for data sets in Geo that deal with the age of a patient, you're interested only in human specimens from patients of a certain age, well, you're in trouble. Because if you go to Geo, you find that when you look at the metadata, you see age and age and age and pop age and age after birth and age in years and all the other variants. And if you're a guy like Pravesh and you want to search for patients who have a certain age range, you have to figure out how could investigators have described the age of their subjects? What are all the possible typos they could have introduced when putting in the age of the patient? And it's just really, really hard. And so what we really need is to make the data more normalized, more standardized, more searchable. Basically, we want to make the data fair. And this is the buzzword that's been sort of permeating the data community for the past 5 or 6 years. The idea that data are going to be findable, accessible, interoperable, and reusable. And it's sort of become almost a game to say that my data are more fair than your data. But making data fair is actually really, really tough because frankly, the tools that most people have for making data sets fair are pretty primitive. And in order to make data fair, they have to be standardized in ways which make them findable and accessible and interoperable, which is just very, very difficult unless you actually have the right kind of computational infrastructure. So what do you need to make fair data? Well, first of all, you need standard ontologies to describe what exists in a data set completely and consistently. And the good thing about biomedicine in general and neuroscience in particular is that we just are really good at coming up with controlled terms for the things that are important to us. Look, Linnaeus came up with a set of control terms for speciation 400 years ago, and we've been doing it ever since. The gene ontology tells us everything we need to know about biological processes and where those biological processes take place within cells and what molecules are responsible for orchestrating those biological processes. We're in good shape there. The Neuroscience Information Framework, which came out of work that was organized by UCSD involving lots and lots of people in the neuroscience community, provides ontologies that are really, really good for describing the kinds of things that are important to folks in neuroscience. And these kinds of terms are obviously valuable when we want to deal with things that are much more specialized than what we could learn from just the standard kinds of ontologies. I should put in a plug and say that we at Stanford have a resource called BioPortal, bioportal.bioontology.org. We have a repository basically of all the world's publicly accessible biomedical ontologies. And so if you're in need of a biomedical ontology for creating metadata, you can go to BioPortal. And because there's actually an overwhelming number of ontologies that people use in biological research, the good news is that BioPortal provides something called an ontology recommender service, which when given information about the task that you have at hand, will basically make a recommendation as to what might be the most appropriate ontology to use for that particular task. So I encourage you to go check it out. So one of the things that BioPortal allows us to do is to get access to the controlled terms that we could use in metadata to make those data more fair. But we need more than just having terms that come from controlled ontologies and controlled terminologies. We need to describe the experiments that we've done completely and consistently so that somebody who looks at the metadata can actually figure out what it is that we did. And the good news there is that we understand what we need to do. We have to have metadata that will tell us what the data are about. They will tell us whether we can search for additional data that we might be interested in that are like the data set we're looking at, that might help us group the data into categories. That's all good. And furthermore, we know that in biology, there's been a strong tradition of trying to come up with metadata standards that will ensure that our metadata descriptions are as complete as they can be to help us figure out what our experiments show. The trend started about 20 years ago when publishers said, well, look, DNA microarrays are a great technology. We're really excited about what they can show in terms of gene expression. But we're not going to publish the results of 25,000 genes over time in our journal. You have to put them in some database. And that was all good. The microarray community recognized that in order to make sense of such metadata, there had to be some discussion in the metadata of what was the substrate of the experiment, what was the platform, what were the experimental conditions. And they came up with Miami, the minimal information about a microarray experiment. And Miami sort of paved the way, saying, look, communities of practice in biology are all interested in making sure that they can understand one another's results. And in order to do so, these are the kinds of minimal things that metadata need to talk about in order to figure out what somebody has done. And so Miami was just a major advance. And then suddenly just everybody got on the bandwagon. So there are information models like MIATA, minimal information about T-cell assays, or MiniMES, minimal metagenome sequence analysis standard, or MISFISHI, and of course BIDS, which those of you who heard Russ Poldrak's talk in the previous session know a lot about already. And so one of the things of importance to us is that the resource that the University of Oxford maintains, called Fair Sharing, has over 100 of these minimal information model standards that have been promoted by individual research communities, all in biology, all of which provide information about the kinds of metadata that need to be specified for people to make sense about what people have recorded for their experiments. That's great. So if you have ontologies and if you have minimal information models, that's really good. But in order for this to be a successful kind of enterprise, we have to make it palatable, maybe if we're lucky, make it even fun, for investigators to be able to describe their experiments completely and consistently. And that's what Cedar is all about. So in Cedar, we have an ecosystem where scientists perform experiments and generate data sets, where there are community-based standards that come out of groups like the microarray community-based standards like Miami, that can define the kinds of requirements for annotating particular kinds of experiments in a uniform fashion. Cedar users will create metadata templates based on these community standards, and they'll fill in those metadata templates to create controlled, normalized metadata that are searchable. And they store Cedar allows investigators then to store their data sets and the accompanying metadata in whatever repositories are going to be relevant. So the approach basically can be viewed as authoring metadata templates. And fortunately, most mortals don't need to worry about that. That's a process done by people who really understand the metadata well. They will annotate their experiments by filling in the templates with appropriate metadata, and then the metadata and the data sets get put in the appropriate repository. One of the things that Cedar does also is that it's able to reuse the data sets it's able to reuse the metadata that have been previously acquired through Cedar, because investigators are able to, Cedar is able to help investigators by taking advantage of previously entered metadata, looking for patterns in those metadata, and using those patterns to make it easier for the investigators to actually fill out their metadata. And I'll show you what that looks like in a sec. So here's what Cedar looks like. I'm going to show you a demo in a moment, but it's kind of easy right now to show you in PowerPoint. Here is the library that a user would see of metadata templates, building blocks for metadata templates, and actually completed, filled out templates. And we want to look at a particular template. Let's look at biosample human. That's a template for filling in the biosample, creating biosample descriptions that go into the biosample repository stored at NIH. We want to populate it. This is what a Cedar template looks like. It's a set of attribute value pairs. We can see that in this situation, the sample name is 56, in this case a number, the organism is Homo sapiens, the tissue is skin of body, the sex is male, and so on. And the idea is by providing a template that corresponds to the expectations, in this case, of the biosample repository, users are better able to create the data sets that are the metadata descriptions that will annotate their data sets in a way which is consistent and standardized. And one of the things that Cedar does is it talks directly to that bioportal ontology repository that I mentioned earlier. So because we have access to BioPortal, we can say, well, let's enter the tissue, and the tissue is going to be taken from the Uberon ontology. So Uberon is an anatomy ontology that also enumerates various kinds of tissues and vertebrates. And we can make a selection from the drop-down list of the kind of tissue we want to specify. And the good news here is that because we've already stored previously entered metadata values, we know that in about half of the previously entered metadata, the tissue was blood, at least if the organism was Homo sapiens. And so we can sort the entries in the drop-down list so that the most frequently used values are going to be at the top, which makes it really easy for a user to specify the source, in this case, of the tissue, because the drop-down list is already organized in a way which makes it really easy to make a selection. Suppose we specify the tissue as lung, and we want to specify what the disease is of the patient from whom the tissue is taken. Well, again, because we have all these previously entered metadata cached, we can know that when the tissue was lung and the organism was Homo sapiens, for example, the most commonly entered value for the disease was, in this case, lung cancer taken from the disease ontology. And chronic obstructive pulmonary disease was number 2, and squamous cell carcinoma of the lung was number 3, and so on. Again, we can sort the entries to make it really easy to specify what the corresponding disease is. And also, if we don't see what we want at the top of the list, we can search for it using the search bar. And so if we say, for example, that the tissue is brain, well, then we're seeing terms like Parkinson's disease and CNS lymphoma and autistic disorder and melanoma. We're not seeing things relating to lung, we're seeing things relating to the nervous system. And so we can see that by taking advantage of the previously entered metadata, Cedar makes it easy. And I think going back to the slide that Nici showed you with the need to be able to make it so that the early adopters can become the foundation for the use of new technology, but eventually things become more normative as communities adopt technology. We really want to make it simple and easy so that we can see increasing uptake of this kind of stuff, so that fundamentally we're not dealing with the cumbersome task of filling in detailed spreadsheets or trying to figure out ontology terms by hand, but rather making it all possible through this kind of technology. And so Cedar basically allows you to enter the template through a template authoring system, filling in that template, using the template to store stuff in a remote repository. What I'll try to do, if things work in my favor, is to show you what this all looks like operationally. Even though I'm realizing we're getting short on time, here's what the Cedar looks like. It's all done through a web-based interface. Here's my Cedar library, which right now has nothing in it. I don't have anything that I've actually entered right now in my library, but I can see what other users have shared with me. And not surprisingly, my colleague John Graybeal has shared with me information from BIDS. And I can just show you very quickly, if I'm interested in BIDS, the biomedical imaging data structure, I can take a look at a metadata description and see what that looks like in Cedar. And I double click on it, and I can see what I have are all the fields that are in BIDS, the dataset name, the BIDS version number, the dataset interpretation type, and I can fill in these values. And in the case of BIDS, everything is a string, so everything is basically entered by hand. But I've entered, for example, just to show you how Cedar works, to supplement the idea of the license for the data set, a licensed unique identifier, to give you a sense how Cedar allows you to go to drop-down menus where I might say, well, if I don't want CC0 to be my license, I can say that I'm using artistic license, and that's okay. I can see that my authors include Paul Broca and other notable neurologists. I don't have to necessarily accept that or believe that, but you get a sense for how the data entry system works. The thing that's really nice about being able to enter information through Cedar's system here is that obviously things that are strings are entered just by type in, but I do have the ability for things which I can define either in terms of predefined data sets or ontologies, getting access to those data sets really easily. What I'll also show you really quickly is that this little symbol here indicates that I've enabled OpenView for this particular metadata element. What that means is that I can publish my metadata as a web page. And so anybody can access that web page. So let me just show you what happens when I go to OSF. Since Nici created an account for me in her particular platform for the Neuromatch Academy, I can go to a document that we've created here that you can all get access to, which includes just some stuff from the README file for one of the entries in OpenNeuro. NeuroOpen rather, but one of the things that I can also see in this README file is that the user has said that the Cedar authored metadata is available here. This is actually not the metadata for this particular entry, but to show you how OpenView works, I can just click on that link and I can see in a web page the information that we just entered into the metadata template in Cedar. And so one of the real advantages that we have right now is that information which we enter into Cedar is publishable on the web. And not only is it publishable on the web in general, it means that we can paste those URLs directly into OSF. And so OSF right now already have a kind of integration through the URLs available through OpenView, the Cedar capability. But we also have the ability, of course, to be able to look at them directly in OSF and take advantage of integrating the two techniques this way. Eventually, we're going to have a much more comprehensive integration of these two technologies. So going back to my slides, let me just say in closing that we don't have to deal with spreadsheets when we're dealing with Cedar. We have really structured, community standard-based metadata templates. We don't have to worry about confusion about how information is going to be entered. We use terms from standard ontologies to make sure that everything is compatible. It means that by having good metadata, our data are fair. It means that if we want fair metadata, frankly, we're not going to be able to do that without metadata that uses common templates based on standards. We're not going to have fair data until those templates can be entered using controlled terms. And fundamentally, we need technology that will make it easy for investigators to annotate their data sets in standardized, searchable ways. So I think Cedar and OSF are addressing complementary problems that are really important for open science. As Nici showed you, OSF is really fantastic at helping investigators to manage their workflow, to register their studies, to share files and data. It really addresses the element of open science that's necessary for rigor and reproducibility. What Cedar really focuses on is helping investigators to annotate their data in standardized ways with standardized templates and standardized terms, which are really essential for making data sets findable, for re-exploring data sets the way Pravesh Khatri has done, and for integrating my data with your data so we might take advantage of economies of scale. And one of the things that we're really excited about is the idea of trying to bring these two technologies together so that we can really show the importance and the advantages of open science in a very broad way. And that's something which I hope we can show you in some future talk. So we really want to thank you for your attention. It's just really exciting for us to know that we can bring COS and Cedar potentially together. They solve really complementary aspects of the open science problem. And by the way, you'll also get a link to a survey that Nici and I would love you to take, which will give us some more information from you about your thoughts about the two technologies and hopefully to give us some advice on what you'd like to see us do moving forward. Thanks so much.
Great, thank you. Thank you. So I've put the link to the survey as the little button on the bottom there. So if you want to click that and go take the survey, that would be great. It sounds like we'll be able to get some good feedback here about how these two tools can work together. So thanks very much to both of you for sharing these exciting tools with us. And I'll echo also what Patrick said in the chat earlier, which is that the Open Science Framework is actually hosting all of our data sets that we're using for the projects and for the tutorials. So we're really grateful to the Open Science Framework. And so what's funny is that, I did this poll at the beginning where I asked the audience, have you used OSF? And a lot of them said, no, not yet, but I'm excited to learn. And the secret is that actually you all are using OSF already. You just don't know it. It's all there. And so we're already gratefully taking advantage of these tools. So your answer is actually yes, whether you knew it or not. Okay, so this is great. So I think we've got a little bit of time for questions, actually. So I know that Nici, you were answering some of the questions in the box, but maybe we could start with the most upvoted question here and talk a little bit for both of you guys about when labs, so I'll just read it out. So it's for the recording here, but when labs share their data, what do they share? Is it raw data? Is it post-processed data? Is it only their results matrices? Is there a general rule of thumb for this? And so, Nici, you said you've seen all of them, but maybe we could all talk about, what the heterogeneity is that you see.
Well, I think the right answer is it depends. And it depends mainly on what the expectations are of people who are going to receive the data or who care about the data. If you're NIH and you funded a project that requires data sharing, then they want to see the data. They want to see the metadata. They want to see everything. If you're a publisher, you may expect that a journal article will be accompanied by links to data that, again, include everything. And so in that case, you're going to be very, very complete. Many people will put data in repositories that are not nearly as expressive. People will use, one of the things that OSF is good about is basically handling data at all of those various levels of granularity. So it's really a function of what the investigator wants to do. But certainly in the case of situations where data sharing is mandated, almost always it means both metadata and data.
Great. Nici, did you want to add?
No, I think Mark's answer was fantastic. He nailed it.
Great, cool. So thanks, Dilce, for your great question. So we've got another question. Actually, I kind of cheated here. I asked this question, but the reason was that I did a poll where I said, like, have you pre-registered a study? And so if we go to the poll's answers, we see, have you pre-registered a study? Nobody said yes, many. Nobody said once. A lot of people said not yet. And a couple people said, what's pre-registration? And so I think that it's really important to talk about this and why we would want to pre-register and how the metadata curation and the open science framework could potentially work together to facilitate that pre-registration, which is such an important aspect in open science in general. So you guys want to jump in?
Yeah, it's a great question. We could talk for another hour, I think, on pre-registration, and I'm sort of sorry I didn't get to spend a lot of time on it in that overview, but in a nutshell, it's simply specifying your research plan in advance. And so there's a lot of ways that could actually be applied, but I think in a nutshell, it's your study design. It's preventing some of that bias that can happen when you collect your data and then decide what you're really going to ask of the data. So this is just sort of doing all of that up front. And I definitely get the question a lot of times, well, what if I do this and then something goes berserk and I need to do some different steps in my process? And that's not a reason not to do it. It's going to happen. It happens to everybody. So doing a first pre-registration, and then if you need to make a change or an update, doing another registration or doing an amended one where you can specify those changes is certainly okay, because I hear that all the time, and that's sort of a scary thing. I think I also mentioned sometimes people need, or for various reasons, need to keep that private for a little bit of time. And that's certainly okay, at least on the OSF. There's other tools out there too that you can use for a registration, but up to 4 years or public immediately. What's also something I didn't mention is that you can get a view-only link to those registrations, and you can anonymize those. So if you wanted to send this on for peer review in some of the different journal circles to get some feedback on your study plan, your study design, you can do that without giving away who you are if you needed to. But nonetheless, it's very simply defining what you're going to do before you collect the data. So maybe that's a good start. And I don't know if you have follow-up, Megan, on that.
Well, I can also let Mark follow up if he wants to.
Yeah, I'll just add that it really is a mechanism by which investigators can go on the record regarding what they're going to do and prevent cheating, prevent p-hacking, and all the things that we're trying to avoid. If you're doing clinical research, if you're doing a clinical trial in the United States, basically you're not allowed to do investigation on human subjects unless you pre-register your study, because it's considered so important to protect human subjects by having the goals of the experiment defined in advance and not violated.
Yeah. We've actually got a couple other follow-up questions about pre-registration, so maybe we should do all of these at once. I'm just going to lump them all together. We've got, are all research questions suited for pre-registration? And then there's a new question that just came in from Homero, which says, I've heard that pre-registration as a way to make clear what were the hypotheses that motivated the experiment. So it's kind of the same thing, like which questions are appropriate. This would make clear when a finding was done after extensive search into a data set, and that decreases the statistical power of the finding. So in the case of theoretical research, this doesn't seem necessary. Am I wrong? So it's, you know, do you have to pre-register theory in addition to pre-registering empirical results, and what kinds of questions are suitable or not suitable for pre-registration?
Yeah, those are great questions. Those are the tough ones, really. And it takes, I think, really getting into the nuance of some of that to understand it fully. My answer in general is yes to everything. So I think that there is the separation between confirmatory research, which is hypothesis testing, and exploratory, which is hypothesis generating. But you can state that sort of in your pre-registration to kind of specify what it is and still continue to do that initial claim at the beginning of what you're going to do. And I think pre-registration fits for both. Theoretical is definitely a little bit harder to sort of break out. And some of the things that I've spent some time thinking on with different communities of researchers is about models. And so some model-based research, we actually are, we have a registration template that we're going to be rolling out later this year that is based on some of that community feedback and thinking about a theoretical model and how you could use it early on in your process by specifying sort of the goals in advance. Mark, I don't know if you have different thoughts.
Great. That's great. Yeah. Mark, no?
Not much to add. I think that was really good. I think even if you're doing an observational study, you know in advance what you're going to observe. And the idea of pre-registration is really to protect the investigators so that no one accuses you of contorting your analyses of multiple comparisons in a way to favor outcomes that you didn't anticipate, but just happen to have a positive p-value.
Yeah, I think this actually ties really nicely into what we're trying to do in Neuromatch Academy too, which is like really think about selecting the tools and analytic approaches and techniques that you want to answer specific questions instead of just trying all of the things and then reporting the p-value that happened to fall below 0.05. Yeah. And so pre-registration fits really well into the theoretical and motivational framework that we're trying to build here, which is choose the right tool for the job and think about why it's the right tool to answer the questions with the goals that you have in mind. So doing a pre-registration actually kind of helps you think through that entire process before you even go and do the work. And so that fits very, very well into what we're trying to do here. So pre-registration is good. So I think the last thing that we have time for here is on the flip side. I'm gonna change Miriam's question a little bit because it kind of goes with what we've been talking about. So Miriam is asking, sharing data requires a lot of work, and I would also add that pre-registration also requires a lot of work. And we've talked about the pros and cons of pre-registration a little bit, but can we expand on, for a postdoc or grad student already under a lot of pressure to be very productive in a very short amount of time, what advantages are there besides avoiding people accusing you of p-hacking or something similar? What kind of concrete advantages do pre-registration and open data and data sharing offer?
Well, I just said that obviously you're protected when you pre-register because no one can accuse you of actually going beyond the initial experiment you had in mind or chasing hypotheses that no one would have imagined previously but happen to fit the data. So that people have confidence your data are not being described in a way which is overfitted. In terms of sharing your data, I think increasingly we're seeing people getting academic credit for sharing data. I think people increasingly are being recognized for their work through the analyses that other people do. I mean, I think this is just part of what the community is and how it's evolving. Yeah, I know there's a lot of concern about putting data out there and what people might do with someone else's data, and I think those are probably overblown concerns. I think there's a lot of discussion about what it would mean for appointments and promotions committees at universities to be using data publication as a criterion for appointment and promotion, but that's happening already, and more of that's going to happen. I think it's just part of being a good scientist, and I think we all want to do that.
Yeah, I would echo everything Mark just said, but I'd also just add, yeah, I agree. I think some of the incentives are shifting. I'm not saying we're seeing a huge change yet, but I definitely am seeing some positive indication of the incentives around the sharing of data and the reuse of data as a positive signal towards somebody's career or areas of research and the way that they conduct it. And so I think for that reason, starting your career as a postdoc or early on in your life, realizing the value of that and beginning to make it habitual would be, I think, at the end, beneficial. I also think, that statement I made earlier just about begin with the end in mind. I think, if you aren't thinking about these metadata pieces, pre-registration, which all take place before you're really doing the data collection and the analysis and then the reporting, it makes it really hard to do that easily in an open, transparent way, which then people question the credibility of things when they don't see the transparency. So I think by doing that from the beginning, you're really adding that value, and you don't have to think or work so hard at it because you were doing it the whole time. You have the version histories, you have all those pieces. And again, the end in mind typically is publication. Again, incentives seem to be still aligned there, and I don't think that's going to change in the near term. And one of the studies that we recently paid attention to was the fact that by citing the open data in a paper, it actually increased the citations of a publication. So I think in that sort of spectrum, there's a lot of benefit, and I just named 3, but I think there's a lot of benefit in thinking about this now at that point in your career.
And let me just add that by the end of your career, I would certainly expect that the norm is going to be that publication is going to take place in terms of data sets and metadata that describe those data sets. That the publications in journals are going to become increasingly irrelevant. I like to say that nobody reads journals now anyway. That's probably overstatement. But eventually we want to have our data out there, not only so that we can have access to the data, but so that computational agents can go out to the internet, find data sets, find information, bring it all together, tell us what we should be knowing, tell us how to plan our next experiments based on what's out there. That's all going to require a new kind of publication. And I think that's the kind of vision that stems from the technology that Nici's group and my group are building. Because really, we don't see publication as a bunch of PDFs out there, but really we see it as data.
And that is a great note to end on. This is the future of science, is data sharing and data access and data curation so that others can learn from your work and build on what you've done. So I think that's a fantastic place to stop. So thank you very much to Nici and Mark for sharing your